Hey guys, welcome to this BrickBio video. My name is Emily, and in this video we will be going over the organization of the nervous system in vertebrates, specifically humans. Let's get into it. Let's begin with some of the properties of nerve cells and how it can conduct these impulses. The inside of the cell is going to be more negative than the outside because at rest there are more potassium ions that leak out than sodium ions in the cell. So how does this work when both ions have the same positive electrical charges? Well, when potassium leaves the nerve cell, there are plenty of membrane proteins that have a negative charge that cannot leave the cell when the positive potassium ions flow out. Boom, negative inside. Anyway, nerve cells have a variety of membrane transport proteins to facilitate the electrical reaction to a stimulus. This includes the good old sodium-potassium pump, which pushes ions up their gradient with active transport, along with leakage and gated channels, which use passive transport to move ions down their gradients. Take note of the ions seen in these processes. The two featured here are sodium and potassium. The sodium-potassium pump is an active transport system that requires ATP to function. When activated, the pump will transport 2 potassium ions into the cell while forcing 3 sodium ions out. When sodium ions are released from the pump, conformation changes occur to be able to transport the potassium molecules, resulting in the dephosphorylation of the protein, and affinity for sodium increases again. As mentioned earlier, the nerve cells also have leaky potassium channels that diffuse it out of the cell and down its concentration gradient. These are also always open. This will be the key to establishing the resting membrane potential. At rest, a typical resting membrane potential will be about negative 70 millivolts. This is regulated by the flow of sodium and potassium ions. However, this can be altered through the presence of a stimulus. The process of making the membrane potential less negative is called depolarization, while hyperpolarization will make the membrane potential more negative. Think of it as a membrane becoming more D-negative during a depolarization, while something that is hyper has more energy, just like how hyperpolarization makes things more negative, if that makes sense. A stimulus will also activate gated ion channels, which are usually closed at resting potential. The opening of these channels allows ions to flow in as they please, which changes the charge of the membrane potential. Thanks, stimulus. There are actually 2 types of these channels. One requires a ligand or neurotransmitter to open, and are called chemically or ligand-gated ion channels. The other responds to changes in the membrane's electrical charges, and are called voltage-gated ion channels. These short disruptions in the membrane potential add up to what we call an action potential. To trigger an action potential, a nerve cell must be depolarized enough. Usually this value sits around negative 55 millivolts for mammals, and is also known as the threshold potential. Action potentials are an all-or-nothing event that won't trigger if this threshold value isn't reached. Crossing this threshold opens the voltage-gated channels for sodium and potassium. Let's take a closer look at this process. Everything is so calm here in the axon hillock. Before that rude stimulus interrupts us, let's discuss the 2 voltage-gated channels. For sodium, its channel has 2 gates, an activation and an inactivation gate. During this resting phase, the activation gate is closed while the inactivation gate is open. Pretty self-explanatory. Potassium's channels only has 1 gate, and that is closed during this phase. At this point, there are plenty of sodium ions outside of the cell, and more potassium ions inside. Oh no, a stimulus. Oh no, it has reached the threshold. Oh no, we're moving down the axon. During this rising phase, the sodium activation gate opens, which floods the inside of the cell with sodium in a steep depolarization. This gate will eventually close at the top of the curve, opening the potassium gate. The potassium ions flow out of the cell, rapidly hyperpolarizing the cell to the point under the threshold, where some excess leaves the cell, causing a slight undershoot. This is also known as a refractory period, where the sodium-potassium pumps have to restore the original flow of things before being able to accept another sufficient stimulus. The sodium activation gates close and the inactivation gate opens. For the potassium gate, the one gate closes. Altogether, this process takes about 1 millisecond. So how does this travel down the axon so fast? Yeah, propagation, basically. Action potentials can only travel in one direction through the nodes of Ranvier via saltatory conduction. Each segment of the axon is depolarized by the action potential as it travels through. You can increase the speed by increasing the amount of myelination or the axon diameter. Let's move on to what happens after the action potential gets to the end of the axon. At the end of neurons, there are regions that collect the signals from action potentials, called synapses. The one at the beginning is called the presynaptic terminal, and the one at the end is called the postsynaptic terminal. These don't physically touch, but are instead separated by a gap called the synaptic cleft. There are also two types of synapses, electrical and chemical. I'm only going to cover chemical synapses for now, which use neurotransmitter binding to ligand-gated channels to control the flow of certain ions. Let's take a look at this process. Just letting you know, the ions here are going to be different than before. Here, we will see the release of calcium. After an action potential reaches the end of an axon, the voltage-gated calcium channels open, releasing a ton of calcium into the terminal axon branch. This then triggers the fusion of synaptic vesicles full of neurotransmitters into that empty synaptic cleft, a process called exocytosis. The frequency of action potentials can also affect the number of neurotransmitters released. A more frequent signal will release more neurotransmitters. There are two different types of neurotransmitters that I'll explain in a second, but I just wanted to add that existing neurotransmitters need to be removed before new signals can be transmitted. This is primarily done through the reuptake of neurotransmitters by presynaptic cells, or enzymatic digestion. An excitatory postsynaptic potential, or EPSP, is going to cause a depolarization, as the binding of these neurotransmitters open the ligand-gated channels to release a ton of sodium ions. Examples of excitatory neurotransmitters include acetylcholine, dopamine, and epinephrine. The depolarization influences the postsynaptic neuron and is more likely to generate action potentials. The opposite of an EPSP, an inhibitory postsynaptic potential, or IPSP, will result in a hyperpolarization of the membrane. This is because the ligand-gated chloride channels are opened, releasing a ton of negatively charged chloride ions into the cell. Examples of inhibitory neurotransmitters include GABA, serotonin, and glycine. Together, EPSPs and IPSPs create a graded potential for the neuron and influence its ability to fire an action potential. There are two types of this summation. Spatial summation is when multiple dendrites contribute to this ability, sort of like an average consensus. Temporal summation is when a single presynaptic dendrite influences this ability by repeatedly firing EPSPs at a rapid rate to depolarize the postsynaptic cell enough to reach the threshold. Phew, that was a lot. Thanks for watching this brief overview of the action potential, and good luck with your studies.